Welcome, everyone, and thank you for joining us today for this forward-thinking discussion on biological collections, organized by the American Institute of Biological Sciences in partnership with the Natural Science Collections Alliance and the Society for the Preservation of Natural History Collections. My name is Jyotsna Pandey, and I'm the Public Policy Director for AIBS and Executive Director for NSCA Alliance. Before we get started, I'd like to make a few housekeeping announcements. This webinar is being recorded. As backup, the webinar is also being live streamed privately on YouTube. The YouTube video is currently private, so it's not searchable or publicly available. And the webinar recording will be shared with all webinar registrants in a follow-up email in a few days. You can use the chat feature, which is at the bottom of your Zoom screen, to communicate with people on the call. If you're experiencing any sort of technical difficulties, please reach out to AIBS IT support, that's Diane and Joel, over the chat for assistance. And if you have any questions for our panelists during the course of the webinar, please type it into the Q&A box. And you can find the Q&A box by clicking on the Q&A icon at the bottom of your Zoom screen. In terms of schedule for today, we have a series of presentations planned for you all, which will be followed by a Q&A session at the end at about 3:30 Eastern. We will aim to take a short 10 to 15 minute break at around 2:45 p.m. Eastern. And that's sort of all the housekeeping sort of announcements. And to get started, today's program is a joint session, a joint discussion between representatives from the National Academy Study on Biological Collections and BCoN's Extended Specimen Network report. Next slide, Andy. Thank you. We have an esteemed panel of speakers joining us today. BCoN, or the Biodiversity Collections Network, released its NSF-funded report envisioning the Extended Specimen Network back in April 2019. We're joined today by members of the BCoN Advisory Council, including Dr. John Bates with the Field Museum of Natural History, who's also the current NSC Alliance President, Mr. Andrew Bentley with the Biodiversity Institute at University of Kansas, Dr. Linda S. Ford with Harvard Museum of Comparative Zoology, Mr. David Jennings with IDIC Bio and Florida Museum of Natural History, Dr. Anna Monfils with Central Michigan University, Dr. Barbara Thiers with the New York Botanical Garden, and Dr. Jen Zaspel with the Milwaukee Public Museum. Next slide, Andy. From the NSF-funded National Academy study on biological collections, the report for which came out in September of last year, we're joined by the two co-chairs, Dr. James Collins with Arizona State University and Dr. Shirley Pomponi with Florida Atlantic University Harbor Branch Oceanographic Institute. And Dr. Pomponi will join us in a few minutes. She's a little bit delayed. We also have Dr. Manzoor Hasbon, who was formerly at the American Titan Culture Collection, and Dr. Kyria Boundy Mills with the University of California at Davis. And I want to note that Andrew Bentley and Barbara Thiers also served on the Academy's panel. Next slide, please. Today, our panelists will be discussing the common themes and recommendations that emerged from these reports and leverage those to have a discussion about the path forward for biological collections. And just to give you a brief overview of the program, we're going to start with a brief introduction of the two reports, their recommendations, and the areas of overlap between them. Followed by a discussion of the various implementation challenges associated with those recommendations. And then after a short break, we will examine a couple of case studies that are practical examples of the type of integration we're envisioning with the Extended Specimen Network. We will then get into the legal compliance issues, after which we'll touch on the role of collections organizations in implementing the recommendations from the two reports, before concluding the presentations and then heading into discussion and Q&A. I am now going to stop talking so we can hear from the experts. First, I'm going to ask Dr. Collins to just start off by briefly introducing the Academy study, and following that, Dr. Barbara Thiers will say a few words about the Extended Specimen Report and then discuss the common themes. Whenever you're ready, Dr. Collins.
Excellent, thank you, Jyotsna. We really appreciate it. Let me start by acknowledging my co-chair, Shirley Pomponi, who has gotten delayed for a bit. She'll come along in a bit. The committee that put this report together and the National Academy of Sciences staff members who all made it possible. So to give you some context, National Science Foundation asked the National Academy for guidance on a series of questions regarding long-term sustainability of biological collections infrastructure. The committee identified several areas of tension, not surprisingly, within the biological collections community. Collections are diverse, communities surrounding collections often operate independently of one another, and there is just a difference, an inherent difference between living stock collections and natural history collections. So we were charged with resolving a lot of this variation. Each chapter lays out the main challenges and a path forward that the committee identified. The recommendations we offer are to the leadership of biological collections, the biological collections community itself, as well as the NSF Directorate for Biological Sciences. So it's important to keep in mind that for these consensus study reports by the National Academy of Sciences, there's a statement of task that goes along with it. And that statement of task really does guide the work of the committee. So if your favorite topic is missing from the report, it may be that it was not actually included in the statement of task. And so it wasn't part of our charge to cover. The report is intended to launch a national conversation about the future of biological collections, and I can think of no better example of a success than this particular meeting, especially as I see where the panelists are from across the country and even across the world. Andy, if I could have the next slide, please. So as we thought about biological collections, we first realized that infrastructure includes many intersecting components. It's physical space, it's the technologies to produce digital data, and the cyber infrastructure to store those data and make them available. And finally, biological infrastructure includes the staff, the collections managers, the directors, the curators. All of these individuals help make these collections go, and they help make them available worldwide. The committee also recognized that future sustainability and growth of collections depends on addressing 4 interrelated issues that are illustrated here in this cartoon. First is to upgrade and maintain the physical infrastructure and also growth of collections. It's important to keep collections growing. Develop and maintain tools and processes needed to transform digital data to an easily accessible and integrated cyber infrastructure. Recruit, train, and support the workforce of the future. So we have to keep in mind the individuals that we're bringing along in terms of training activities. And that also means that we have to keep in mind issues related to justice, equity, diversity, and inclusion when it comes to the whole idea of inclusive excellence and diversifying our community. Finally, there's the whole idea that I don't have to remind this group very much of. It has to do with ensuring long-term financial sustainability when it comes to these real important and wonderful resources. Now, realizing the committee's vision is going to require enhanced communication and collaboration within the collections community. And that's where we get to the next slide, Andy, and a couple of ideas that the committee had. So in thinking about the challenges of the collections community, it goes beyond any single collection. There's growing recognition that an integrated response is something that can be very, very valuable. Several research communities have used these kinds of integrated responses, multi-tiered networks, bringing together individuals of different sorts and from different places. And so 2 things that we recommend, and will be important, I think, by the time we get to the end of this conversation, is the notion of an action community for biological collections to coordinate action and knowledge. Action center related to biological collections, it's an open term. It's one that can be changed. The other thing to keep in mind is the possibility of NSF leading an effort to develop a vision and strategy, such as a decadal survey. Other areas of science use this, physicists, astronomers, oceanographers, and it's the kind of broad scale integrative thinking that could be really, really valuable for this particular community. So NSF then should expand its partnership also across a variety of agencies, across a variety of directorates within the NSF itself in order to advance biological collections. And with that, I'm going to stop and pass on to Barbara.
Okay, thanks, Jim. To say a few words, just to remind you about the Extended Specimen Network report, which some of you have heard about before. This was an effort that was led by the BCoN Advisory Committee. And it grew out of a year-long consultation with the biological collections and research communities. And this whole effort was conducted at the request of the National Science Foundation. It culminated in a workshop in October of 2018, where 20 community representatives pulled all the information together and crafted the report. The report argues that transforming our wealth of physical specimens and related digital data into an extended specimen network should be the next community priority. To realize this vision will involve additional collection and making of new collections and digitization of specimens and derivative data, developing a more robust means of linking data from the primary specimens to these derivatives, creating enhanced portals to increase the utility of these data for a wider range of research and education, and sustaining the network through the development of a data literate workforce and a permanent repository for the specimen data. Next slide, please, Andy. Although the goals of the National Academy and BCoN reports were somewhat similar, the recommendations of these reports were somewhat different, I meant to say. The recommendations by these reports overlap in some key areas, which are summarized on this slide. All the recommendations of both reports could deserve consideration, but in figuring out how to move forward with implementation, we think it makes sense first to focus on those areas of overlap. Although both the NAS and BCoN reports were funded by the National Science Foundation to provide advice on the future of US biodiversity collections, we want to emphasize that harnessing the full power of biological community collections will require an international collaboration. We have a lot to learn from our colleagues in other countries, and we're committed to making sure that our activities are in alignment with theirs and helping to build capacity in other countries where that's appropriate. Others will say more about what we're doing now in terms of international collaboration. And so I'll turn it over to Andy now.
Thanks, Barbara. So it became very obvious to us that there were lots of commonalities in the recommendations between these two reports, and there was a shared vision for the future of collections. One of the things that we did is we looked at all of the recommendations, and we noticed that they all fell into sort of 5 broad thematic areas, or what we sort of colloquially called pillars, that were required to implement this extended specimen network concept. And those fell into the areas of collecting. We obviously need to continue to collect in order to grow our collections. We need to continue to digitize and get that information up and online. We need to improve the infrastructure associated with our collections. We need to integrate all of this information together in order for us to be able to answer these big questions. And then we also need to improve the workforce and education associated with those collections. And so there are a number of ongoing efforts that are putting all of these pieces in place. Some of them have been implemented, but there are some challenges and some holes that we need to continue to fill. So what I'm going to do is move on to the first of these. We're going to go through each of these broad thematic areas one at a time and discuss them in a little more detail. And I'll start with the collecting one. We obviously need to continue to collect. We need to continue to grow our collections. We need to improve our vouchering, and we need to improve our ability to answer big questions by using the temporal series of these specimens that we have in our collections. We also need to do more holistic collecting, not only just collecting our target taxa, but also collecting multidisciplinary collections while we're out in the field. There are obviously some challenges associated with that. We also need to collect multiple preparation types while we're out in the field, collecting tissues, collecting parasites, so that we can answer these host parasite, plant pollinator, tissue voucher kind of relationship questions associated with our collections. We need to promote collaboration with collections before researchers even go out into the field, so that they can identify gaps, taxonomic gaps, geographic gaps, and temporal gaps, in order to improve the scope of our collections to answer these questions. We need to also ensure that these collections are born digital, to alleviate some of the pressure that we already see associated with our collections, and make sure that that information is very easily incorporated into the collections once it gets back from the field. And then we also need to incorporate observation information. There's a lot of observations that are made by collectors that don't necessarily make it out into the world to be used to answer these kinds of questions. There are obviously a number of challenges associated with collecting. Obviously, there are new permitting restrictions that are restricting the amount of international and regional collections that we can actually undertake. Collecting priorities are driven by research dollars, and there is little direct support from collections from research. So one of the things that we have proposed in the Extended Specimen Network report is the implementation of a specimen management plan, analogous to the data management plan that needs to be done for all research proposals going through NSF. We are proposing something analogous to that that will link the research dollars to the collections maintenance. Dollars will be associated with a research proposal that will support the digitization, the curation, and long-term care of any specimens that are collected along with that research. A dollar amount per specimen will be added to the research proposal that will then be able to fund that digitization, curation, and long-term care. There needs to be a collaborative agreement during the proposal phase, identifying a voucher repository where those vouchers will be deposited, and ensuring that the collection is willing to take those specimens on. This will also then obviously reinforce the value of voucher deposition. So I'm going to hand over to David Jennings now, who's going to talk a little bit more about digitization and dark data.
Thank you, Andy. So obviously, especially if we think about this past year, it demonstrated how the physical objects and digital objects have really helped. So what we need to do is continue leveraging our ongoing investments in digitization efforts. We've made huge progress, but yet by some estimates, we've covered maybe about 30% of specimens in collections that are digitized. So there's this huge untapped potential for digitization. And so a lot of that comes down to there are some collection types that are very hard to digitize relative to others. For example, entomology and paleontology. So we need to continue our advances in the technology for doing the digitization so that we can make progress. Also, we need to look and find the dark data, this data that exists, but we may not know about it. There's small collections, there's project-based collections that can provide really rich views into biodiversity of areas, and we'd like to find those and get those digitized so we can add them into the overall big picture. We need to think about integrating all different kinds of collection types so that we can establish these relationships like Andy was talking about. You know, we're talking about integrating living collections, geological collections, and associated those with the biodiversity specimens. And when we make new collections, you know, we can create those as digital objects from the start. But we also have a lot of other existing collections that we'd like to augment the data and turn those into extended specimens. And that means we would augment the data with things like field notes, images, CT scans, sequences, publications. There's a host of other data that we can associate and turn those into digital specimens. And then also, we'd like to complete like stub records with a focus on research use, like, you know, maybe prioritize how to take those data from, you know, from where they are now into making extended specimens. So some of the challenges with implementing continued digitization, the first one is data cleaning, standardization, and deduplication. You know, various aggregators do data cleaning so that the computer can represent it and provide access, although sometimes that has an unintended effect of creating duplicates that aren't really duplicates. So what we need to do is work on common methods for data cleaning, common methods for how we represent the data so that we have one data set. Also, it would be great to have a comprehensive registry of collections, because this will help tell us, you know, what do we have and where are they, and so that we know what the scope of our effort is going to be. And lastly, I like to think about this as a culture change. We want digitization to become part of the normal curation process, not something that's addition or not something that we have to pay extra for. It should be something that we think is just normal part of the process. So with that, I'm going to pass it back to Andy to talk about the next topic, integration and cyber infrastructure.
Thanks, David. So one of the things that occurred to us very early on is that integration is certainly the next frontier when we're talking about collections. Extended specimens obviously rely on the integration of data. They allow us to answer these more complex and more important questions through the integration of all of these different pieces of the specimens that are associated with them. We obviously need integration at all levels, right from the preparation level where you have multiple preparations associated with a specimen, to integrating various different specimens together, to integrating different collections together, and then also integrating various different data sources from outside our traditional realm in order to be able to augment all of this data and make it possible for us to answer these complex questions. We also need to integrate the products of all of the research that's being done on our specimens. As David said, publications, sequences, isotopes, CT scans, images, sounds, videos, all of those kinds of things in order to make these specimens more useful to a broader swath of people who are now starting to use our collections. This whole system is obviously reliant on unique identifiers at various levels, identifiers for the objects, identifiers for data sets, identifiers for collections and institutions, and identifiers for people. And their use throughout the data lifecycle will obviously be important for us in order to be able to track the connections between all of these different pieces. So everybody needs to play the game. Everybody who has a stake in this or is a part of the data lifecycle obviously needs to play the game and needs to be involved in this process in order for us to be able to make those connections. Obviously, the whole system is also reliant on published, complete, and fair data. We need to get as much of this data up and online and available to be used in order for this process to work. And it also requires robust cyber infrastructure to support not only the digitization of the specimens, but also the storage of all of the information that we're starting to accumulate and the dissemination of that information out onto the web. Integration is also important for collections attribution, annotation, and legal compliance. Obviously, this whole system of data integration will be a mechanism for us to show our worth to the scientific enterprise as a collections community. We can also use it for advocating for collections funding by showing our importance to the research endeavor and the kinds of information that we can supply in order for this broader group of people to be able to answer these important questions. It'll also bring about community engagement and annotation, where we can get the community to annotate the data, clean the data, correct the data, edit the data, whatever the case may be, in order to be able to improve that data for other people's use. It also allows us to comply with a lot of the requirements that are associated with the Nagoya Protocol and various other legal requirements in order for us to be able to track all of those interactions that are happening with specimens being sent around the world and being used by various different researchers. So some of the parts of the cyber infrastructure are already starting to be put in place. There are various initiatives that are looking at putting some of the pieces of this puzzle together. Things like the GBIF Collections Registry and Index Herbariorum and other collections registries that are out there. The new GBIF clustering tool that allows us to associate specimens based on suites of data. The DISCO OpenDS concept is obviously an important part of this process as well. The IGSN sample registry, looking at being able to register individual samples and have unique identifiers associated with them. And then things like the Pensoft Offer writing tool that allows you to then create a materials examined section within your publication that then can be used to associate the specimens to the publications and vice versa. But there are obviously still significant holes and hurdles that we need to try and look at. Some of the challenges, we still have no underlying unifying identifier system that we can use to then bring all of this data together. A registry of those identifiers would obviously resolve that problem and is a potential way forward. The data landscape is obviously very complex and very convoluted at the moment. We need to simplify and we need to streamline that data stream in order to be able to make it more useful and be able to make those connections. We need to identify and fill gaps in the cyber infrastructure. We need to look at where we can fill the gaps, what new technologies we need to implement in order to be able to put this whole picture together. And then we need to implement this technology in order to circumvent some of the current social needs, some of the mechanisms that we currently use to make those connections, relying on researchers to report publications and GenBank sequences back to the collections. We also potentially need a shift in our traditional publishing mechanism to a more transactional system rather than an archival dump of the data, so that we can get at all of these transactions that are being done between the different elements of the collections. And then we need to link our current highly distributed and disparate communities together to try and ensure that everybody is forging forward in the same direction and is all on the same page. So with that, I'm going to hand back to Barbara, who's going to talk a little bit about infrastructure.
Thanks, Andy. As best we can tell, there are probably about 2,500 biodiversity collections in the US, with perhaps as many as 1 billion specimens. These collections include both preserved as well as living collections. My colleague Kyria Boundy-Mills will address living collections specifically in a few minutes. The infrastructure needed to maintain the scientific and educational integrity of specimens varies greatly depending on what they are, but minimum standards for the preservation of each type do exist, and all require ongoing funding and staff to maintain. Continuing the maintenance of the physical specimens is obviously key to the success of an extended specimen network. Next slide, please. Responsibility for funding the maintenance of physical collections falls largely on the institutions that house them. Most of you on this call are probably aware of the effort needed just to maintain a collection, a task that has been greatly complicated by the pandemic shutdown. The best way to ensure that a collection maintains an adequate operational budget is to have a clear understanding of the current and future needs and to document these in a strategic plan whose goals align closely with those of the parent institution. It's important to make sure that collections are used in critical research projects to the extent that you can influence that and are relevant to the institutional, regional, and national education and outreach activities, and be able to document the value of the collection for national priorities using clear metrics. The cyber infrastructure that's proposed for the Extended Specimen Network will provide a means for tracking the use of specimen data and can provide a basis for at least some metrics needed to demonstrate collection relevance. Next slide, please. In addition to individual infrastructure, there's also community structure in the form of a national network of collections, which is as important as cabinetry and climate control to their success as a knowledge resource for research and education. Only by combining our data can collections meet their goal of creating a complete record of life on Earth, past and present. And each collection, large and small, holds a piece of that puzzle, so to speak. There are many ways that collections work together now, usually united by collection type, e.g. herbarium specimens, fossils, insect collections. However, many more opportunities do exist for collaboration based on geographical proximity, temporal focus, or other similarities. What hampers such collaboration at the moment is that we don't yet have a comprehensive source of up-to-date information about US biological collections, let alone a global source. This has been mentioned a couple of times. The structure for such a registry does exist in GBIF's Registry of Scientific Collections. What's missing, though, is populating that registry with up-to-date information on the location, contact, and content of those collections. And we need this both in the US and on a worldwide basis. And also in terms of infrastructure, as has been mentioned, we need a long-term facility that would, for the long-term management of the Extended Specimen Network and associated data repositories and portals, as well as for continuing education and workforce training initiatives. I've shown here the NEON headquarters in Boulder, Colorado, which might be a nice model for a collections action center, just saying. Okay, so now I'll turn it over to Kyria Boundy-Mills.
I want to thank everyone for the opportunity to address living collections as part of this effort. First of all, there's several recommendations from the various reports that we're exploring today that could bring together both natural history museums and living stock collections, including the Registry of Collections, a centralized action center. So I'll start by giving a brief overview of what living stock collections are for two reasons. First, some of these living collections could be more appropriately be incorporated into these plans than others. And second, some types of collections could serve as a model, such as for centralizing various functions. So in this slide, the blue examples box lists many types of living stock collections that are divided into three general categories. At one end of the spectrum, we have the collections whose holdings are most similar to natural history museums, and those are the biodiversity collections. These consist of a broad taxonomic range of wild-type organisms, such as zoos or seed banks. And these are the least centralized types of living collections. And these would be the most easy to incorporate into some of these proposed activities. In the middle of the range, we have the germplasm collections that consist of many wild-type isolates of a smaller number of species or related species, such as crop germplasm collections, which are used to improve crops. At the most specialized end of the spectrum are the genetic stock centers. For 50 years or more, these types of collections have taken that extended specimen concept to the extreme. They consist of thousands of genetically modified derivatives of one species, or even one single original specimen of a model organism. And they're the central repository for the specimens and data that are used by thousands of research labs. So they're very centralized. For example, for E. coli, you can get the type strain of a specimen of that species from dozens of different biodiversity collections. There are a handful of repositories that have thousands of wild isolates, such as those that are used to track an E. coli outbreak on spinach. There is one E. coli genetic stock center in the world, and it's located at Yale. And that is a very centralized repository that serves thousands of research labs. Next slide. So some of the differences between living stock collections and natural history museums will help guide some of the decisions that have to be made. First of all, of course, living stock collections propagate and distribute specimens to the researchers' labs. Second, there are many different types of living collections because they're used in many different areas of basic and applied life science based on the properties of the organism. I've listed a few examples here. The fungus called Neurospora is used for classical genetic studies because the products of meiosis are neatly packaged in these little sets of 8 spores, so they can be used to track genetic changes. And those strains are maintained at the Fungal Genetics Stock Center. NIH supports a collection of this salamander called axolotl, and it's used to study limb regeneration because this species can regenerate its limbs very well. A third major difference is because these different organisms are used by separate research communities, living stock collections do not interact with each other nearly to the extent that natural history museums do. Next slide. Living stock collections have printed catalogs for many decades and launched online catalogs at least 25 years ago because their users need information about the specimens and the relevant data associated with them. The type of data that is published by a collection depends on how that organism is used in research. So different categories of collections and different specific collections within a category will have different types of data. For example, the average age at which a certain breed of chicken will begin to lay eggs, that would only be useful for one certain category of livestock collections. There's some additional features of genetic stock centers that make them useful as a model for the extended specimen concept. In addition to having the specimens and the data, They have an inventoried list of all the genes in the genome. They have a curated data supported gene annotation performed by the community, which submits information to a centralized database. And they also have developed tools which are developed and shared within that research community. Next slide. There are registries of different categories of collections. I've listed a few examples here. There's at the US level and the international level, there are registries of microbe culture collections. There are registries of seed libraries, for example. Also, there are also existing some data aggregators for different types of collections. For example, GRIN Global is a data platform for data associated with crops. I want to specifically mention the Research Resource ID, or RRID. This is a portal that's a great model to consider for this effort because it links specific specimens, the repository where it's available, and the publications that cite that material. Next slide. So this knowledge of the diverse nature of living collections and how they differ from natural history museums is really important to consider to guide decisions on what types of collections to include in which efforts. Thank you.
Sorry about that. So what I'm going to talk about briefly is the idea of education and workforce training. Specifically, as we plan for this future of biological collections and the extended specimen network, we have to plan for the training of the workforce that is going to support this, as well as the end users of the data. And so that's a pretty wide swath, including future biodiversity workforce, the current biodiversity workforce, cyber collection professionals, graduate and undergraduate students in all disciplines, K through 12, and then citizen scientists as well. At the center of the efforts that we are making, we need to think about training and education in the sense of a community effort to also look at the issues and address diversity, equity, inclusion, and justice. The current and next generation of biodiversity scientists, STEM professionals, and educated citizenry will need extended training in order to be prepared to contribute and do research utilizing this extended specimen network. Next. So part of this is what are our next steps, and both reports address this to some extent with 4 main categories, which is definitely important to be training the current workforce and continuing education. The next was training the next generation of cyber collection professionals. And part of that was thinking about our current training, as well as looking at some professional pathways and programs for the cyber collection managers. There's also the idea of training and engaging new end users of the biodiversity data, and that can be happening across many different levels of education, from citizen science to K through 12 to undergrad, and not just those undergrads that might be pursuing biodiversity science, but those who might become affiliated end users. And then the last but not the least is diversity, equity, and inclusion, and working within our societies and across all levels of development within education, looking at restructuring a lot of our education to think in terms of these terms and creating a more diverse workforce. Next. So some of the challenges that we experience is knowing the depth and breadth of the problem that we have in terms of cyber collections workforce. And so we need to do a needs assessment. So that includes a workforce analysis, defining the status of need and opportunity within the field, defining the necessary skills of the 21st century workforce that is developing and maintaining these resources, and then defining training needs and leveraging existing efforts and ongoing efforts. The next is coordinated training in biodiversity and data science at multiple levels. We want to create the workforce that utilizes this data, which requires a different type of training than we're currently experiencing across our formal and informal education. One that has come up repeatedly is the idea of accessible database interface designed for education and outreach. Our current interfaces are designed for researchers, which is serving as an impediment to end users. And looking at those specific needs and identifying them as a community, it has been identified as a need, but would require more of a community level effort. And that last is a community plan and funding to address diversity, equity, inclusion at multiple levels. So first addressing best practices in this area, then making sure that that is across all levels of education addressed in a very consistent way. I think it goes on now to Jyotsna.
Thank you, Anna. So we still have a few minutes to go before the break, which starts in about 5 minutes. So maybe we can take a question or two. So we have a couple of questions that are related to education and training issues. So Dr. Monfils, this might be something for you. Beckett Sterner asked, what plans are currently being developed to document the biological collections workforce, especially in terms of diversity? And a somewhat related question related to workforce development from Deborah Paul. She asked, does workforce and education include recommending development of some professional development pathways or programs? And I know Keegan Sawyer answered the question. She wrote in the question and answered the question in the Q&A box. But if there's anything you'd like to add to that, that would be great.
Are you thinking now in terms of addressing them, or did you want to push that to later?
We have time for a question.
Okay. Yeah. That's a question. And one of the reasons we're all here together is to start thinking of some community responses and potential ways to address those. So I did see Deb Paul's question earlier, and looking at specifically what sort of pathways we might have into the biodiversity sciences, and particularly into the curation. I don't know that that has been identified, but I think identifying those steps, the pathways and the skills, has been noted as an important step. And so Keegan did a good job of answering that. So I'll kind of punt a little to that. But a lot of this is engaging the community and addressing those issues. And then in terms of what are we doing in terms of diversity, I think that we need to do a lot. I'm not sure that we're making very consistent efforts across our community in that way. I know that there are existing programs that different museums might be using or individual educators might be using. And one of the calls that has come through in both documents is to think about this in a much more cohesive way so that we start addressing this kind of at a society level on down.
All right, thank you. I think we have time for one more before the break. And I'll open this up to the whole panel, whoever wants to answer this. Elizabeth Womack asks, shared data cleaning and data standardization is a great idea, but how do you picture that will work across different collection types, such as paleo, herbarium, geology, cultural collections, vertebrate, etc.?
I'd like to take a stab at that. And I think the quick answer is that ways to address it across all databases of all kinds at one time probably aren't going to work. But there are lots of commonalities associated with our databases. And so I think that encouraging groups to start looking at things, the ornithological community is about to have a workshop to work on standardization of some of the specific fields that are unstandardized, even though you'd think they would be perfectly simple, such as sex. And it's things like that that'll help clean up these databases. And as we go from one set of data to another, we can begin to clean up these data sets more collectively across everything that we have.
All right, thank you, John. So, all right, so we're now scheduled to take a short break so that we can all just stand up, stretch our legs, and maybe refill our coffee and teacups. The program will resume in 15 minutes at 3 p.m. Eastern. And on the screen now, you can see a preview of what's to come next. Thank you all, and see you all in 15 minutes. All right, let's go ahead and get started. Hope everyone is back from the short break. Next on our program are, next slide, please. 2 case studies that are examples illustrating how we can use biological collections and living collections to solve bigger problems. The first is about the terrestrial parasite tracker project that Dr. Zaspel will talk about. And the second is about the role of living collections in responding to infectious diseases, particularly with respect to the COVID-19 pandemic, which Dr. Hasbon will discuss. With that, I'm going to yield to Dr. Zaspel.
Thank you. Welcome back, everybody. You've now heard from several of our panelists about some of the current work and progress towards an implementation plan for the Extended Specimen Network. That said, you've also heard about the challenges that remain in each of the 5 pillar or thematic areas we are discussing. Some of these challenges have been brought into focus through our experiences in working on large-scale digitization projects. Although many of the challenges the collection community faces in the context of specimen digitization are prevalent regardless of project theme or taxon group, we are using the Terrestrial Parasite Tracker Thematics Collection Network as an example of how these pillars can be brought together in a single project and serve as a case study, highlighting some specific issues that still require attention for successful implementation of an extended specimen network. Next slide. We felt this project was a good starting point for this conversation because it has a large number of participating institutions and also because the people involved represent multiple disciplines within the biological sciences. Finally, the data that will result from the project has the potential to extend its reach into a relatively unique set of stakeholders, for example, medical and veterinary fields of study and practice. So just a little bit about TPT. This project started September 1st, 2019, so we're about halfway through the project period. We have 28 participating institutions, 34 co-PIs and leads on subawards. We have over 120 participants in this project at this point, which is very exciting. Our primary objective is to aggregate arthropod parasite collections to build an accessible, comprehensive database of parasite host associations and vector distributions in order to provide the needed baseline information for research and management of the ecological interactions among parasites, pathogens, and their hosts in North America. This figure on the right is showing the taxonomic scope of this project. We are focused on digitization of lice, fleas, ticks and mites, biting true bugs, and biting flies. Next slide. This figure is showing a generalized workflow for the TPT project. On the left, you can see the flow from specimens to data to aggregation and finally dissemination of data. No problem, right? Well, if you look to the right of this flow chart, you'll notice we are digitizing specimens stored and prepared in a variety of different ways, requiring different kinds of equipment and workflows for each type of preparation. Sometimes multiple cameras and workflows, depending on the size of the specimen, are required. Labels often have to be removed from pins or taken out of vials and imaged separately. This work requires a lot of resources, both in terms of people with experience in handling this kind of material, and also obviously the equipment itself is expensive. In order to get the data transcribed, you need a taxonomy table or tree formatted for the type of database you are using. This project accepts all platforms. And these require not only current taxonomies, but historical names or synonyms, as many collections are curated under those older names. In the case of parasites, we are often transcribing label data with information about the host too, which also require taxonomy. It also requires knowledge of which species name on that label is the parasite and which is the host. And finally, which data fields the host and any association data get entered into which fields. This could vary greatly by the type of database or the type of interaction. Establishing standards for this process is very important because if you want to be able to find data associated with a particular type of interaction for downstream research, you need to know where to look for that information once it goes out to our aggregators. So there's definitely a lot to consider. I've selected this image here on the right of a tick and its label to illustrate how each of these thematic or pillar areas are vitally important to successful implementation of the Extended Specimen Network. You probably can't read this label, but I will tell you. It says Lone Star Tick pulled from Susan at 6 a.m. on 6/25/07. probably attached herself on 6/24/07 at noon, left Judy's at 9 a.m. driving. Now, at our last in-person TPT workshop, we had 60 people try and transcribe this record by hand during a group exercise. And as you can imagine, there was a decent amount of variation in terms of interpretation and how to handle data like this. So from collecting the specimen and its data to getting it published requires a lot of investment in each of these pillar areas, especially if we want to address questions about harmful species and their pathogens in a comprehensive way. Next slide, please. To that end, we have now gathered a working group focused on biological collections and associated specimen data relevant to global health and biodiversity issues. We see a need for additional resources to provide infrastructure for coordinating efforts across various projects and groups generating specimen data relevant to global health, ultimately allowing us to get a more complete picture of where disease vectors and hosts have occurred in the past, which will inform research and predictions about the future. Please feel free to reach out to me if you have questions or are interested in joining this working group. And with that, I'm going to hand it off to Mansoor.
Hello, I am Mansoor Hasbon. I used to work at the ATCC until last Friday, so that's why I don't have an affiliation in the program. I start a new position next week, so that's why I'm in between jobs. But what I want to talk is a success story of the value and the role of a living collection as a critical resource to respond to pandemics. Next slide, please. So first, I want to show what do we do in a living culture collection? What is the role that we do and why are we doing all of this? And what is the value of that as a response to a pandemic? So the culture collections, we provide qualified materials to the community. So we make sure that the material is pure, is viable, is well preserved. We confirm the identification of the organism. We do quality control. We make sure it's properly stored. We make sure all of this is following the appropriate biosafety guidelines for that particular material. And then not only have all the biological material process done in-house, but we also try to gather as much information for that material to be added into the description of this item. So for that, you have to create a website with all the description there that is searchable with a good description. And this will also allow the community to access the information, find it, and also order it. When you do that, you have to also make sure that you can send the material to the person that can receive the material. So if you are ordering material of biosafety level 3, you need to make sure that that person is compliant with biosafety level 3 guidelines, and it's also going to be safe for that person to handle that material. So this is very important in compliance. For that, we develop a process to register the person, to have a traceability of the material, to have the proper documentation, all the permits to receive the material, or to send the material to us, because sometimes they do deposits into the collection. And not only having all these, but you also have to have developed all the process for shipping the material. So if the material is to be maintained frozen, so to keep the cold chain during shipping, this can be done domestically or internationally. We do it properly. You have to learn how to package, and you need to make sure that all the import and export permits are in place. So if you look at this slide, this is a combination of everything we have spoken today here. We have the infrastructure, we have the proper personnel with the proper expertise, we have all the information and as much information gathered to that particular material available and searchable to the community. We know how to follow the regulations, and we also know how to ship the material. So ATCC here on the left, you have all the nitrogen tanks. It is a huge collection of all types of material and combines all of these efforts. So the next slide, please. So in the living collection, we not only have this material there, but because this material is consistently done, and thank God biological material most of the time can be grown again and sent again, is consistent. So if you order today one vial of one organism, the next one you get will be very close to that one, very similar, and can be used as a standard. So this is very important because these materials can be always used as control and standards for different applications. So we have authenticated isolates. We can adjust, adapt a new strain of a virus to grow in a particular cell line. And these materials can be used for new diagnostic assays, for clinical and preclinical studies. We can provide not only the organism of interest, but near neighbors for inclusivity and exclusivity panels. We can produce not only that material, but derivative materials. So if somebody has a biosafety level 1 lab, can also work with a material from a biosafety level 3 reagent, so we can inactivate the material. We have antigens, antibodies, nucleic acids. And all of that is key to produce and distribute diagnostic assays. The next slide. So here I'm showing an example of the value of the combination of all the work, not only within the collection, but from other collections, and that includes biodiversity collections. Somebody eventually in a museum had a sample of a bat from China from 150 years ago. Somebody did a microbiome analysis and found a coronavirus in there. So all of this information has been combined and gives all the tools for us to make analysis like this. So here we have a phylogenetic tree showing how over time the coronaviruses have been changing different hosts from bat, pangolin, bat, and suddenly they became adapted to human. And if you look those that are adapted to human, you look at the coronaviruses that produce the normal cold and that are less severe infection, but then also looking at the more severe coronaviruses like those that produce MERS, COVID-19 or SARS, they tend to cluster in a different branch in the phylogenetic tree. All of this information is obtained thanks to the different types of samples and sources over time that can be gathered. And this can happen thanks to biological collections and living collections. Next slide. So thanks to the combination of all of these features, ATCC, for example, has been channeled, the government has channeled the response in many ways to the pandemic. So here I'm talking only about COVID-19, but we have done this with Ebola, with SARS, with the Zika. So the government, through different entities, for example, NIH using the National Institute of Allergy and Infectious Diseases, they fund a program called DEI Resources, where we have all kinds of material available for researchers, free of charge, available throughout the world. The CDC funds another program called the International Reagent Resources, IRR, where they channel and distribute, for example, diagnostic assays for different diseases and outbreaks. So here we can produce, develop materials. So the government helps us to find some clinical specimens or some isolates. And also we find on our own other isolates, and we are creating databases and banks for different materials with different viral isolates, genomic and synthetic nucleic acids, inactivated material, antibodies. We characterize serum and plasma that can be used for validation of different diagnostic assays, for example, of that great interest for BARDA and the FDA. So the biological collections are playing a key role in a response, and we see all the time now with COVID-19. Thank you. So now I will have pass the baton to Linda to talk about legal and compliance.
Thank you, Mansour. Today, I'd like to very briefly talk about legal compliance in biodiversity collections and how some of the concepts that have been discussed today, how can they benefit collections on these issues? Legal issues are a substantial concern for collections with our successful digitization. Much of we have an increased visibility, and many collections, especially in the US, originate from other countries, and so legal documentation is required. New legal landscape includes collection movements such as shipments, but also the Nagoya Protocol, which was implemented by the UN Convention on Biological Diversity. Next slide. This is very briefly, not really going to go over this, but just some of the highlights of the Nagoya Protocol. If you're not familiar with it, I would suggest you Google it. There's a lot of good papers that have come out on this in the last year or so. But it came into force in 2014, and it outlines obligations for genetic resources. And that includes traditional specimens as well, because you can get genetic material out of those resources. And another thing that most people do not realize is that even non-signatory countries must comply with domestic ABS regulation. Next slide, please. Now, what are the new obligations for collections. You need to track all your documentations associated with your specimens. You need to share and document the benefits. You need to record all instances of use and allow access to this documentation. Now in Nagoya, you need to do this legal compliance for the entire life cycle of a specimen, and I'll explain that a little more in just a second. But that means maintaining all the documents throughout the use and research life of that specimen. And that's tracking all activities, transactions from the point of collection all the way in perpetuity. Next slide, please. Now, what are some of the challenges associated with this that collections face? Not all legal documents are the same. It means their retrievability that can't be handled sometimes exactly the same way when you're looking for something. It can rank back, thank you. It can be permits, contracts, MOUs, and also rights. Also, there's a temporal component associated with many of these documents. They can be one time, multiple times, or range of time of use. Another issue is that upstream permission agreements are typically done by researchers, but the downstream use in accordance with Nagoya is now the responsibility of the collection. And there needs to be a flexibility for tracking this because the upstream agreement might change through time if somebody renegotiates. Next slide, please. Now, what I mean by life cycle, we know a specimen is collected, it's accessioned into a collection, and I mean here specifically transfer of ownership, it's cataloged, and then it's ready for loan and use, whether in-house or outside. We all track our in-house use, usually through publications, and we can report on those benefits. But with Nagoya, next slide, please, we now have to track the outside use. And that means we need to not only keep track of what the benefits and the use was from that researcher, but we need to inform them exactly what they can do with that specimen in relation to what they want to use it for if it's in compliance with the original documentation. Now, if you have one specimen, you let them know what that one agreement is. If you're sending a couple of specimens from different accessions, then you may need to send multiple agreements. Next slide. But you can quickly see how if you have multiple specimens from many accessions, there's a lot of documentation you need to send to that researcher and a lot that so they know what they can and cannot do. Next slide, please. So what does this mean in conclusion for collections? We have to increase our transparency. We have to find digital solutions because it's not just a matter of archiving anymore. We now have to have easy retrieval for our researchers. And that means compliance documents, transactions, products of use, and also the metadata of the original benefit sharing agreements. And there's some examples on this slide showing you of what some examples of each one of these categories. Next slide, please. So how do we address this complexity? Collections need to find individual solutions for their compliance and tracking and be able to track that within their own institutions. But on top of that, we need to address this on a global scale where we have a networked provenance and existing use agreements. This means data structures and systems allowing for query, storage, retrieval of all the permissions and rights metadata. This increased transparency will benefit the whole community, not only the collections and the researchers using those collections, but also the providing countries. This latter call is an example of an extended specimen network that would greatly enhance collections for research and education use. Thank you. And with that, I'm going to hand it over to John Bates.
Thanks, Linda. Hi, everyone. My name's John Bates, and I'm the current president of the Natural Science Collections Alliance. And I'm going to talk a little bit about the role of organizations in implementing these recommendations, collections organizations. And as you've already heard, and as you well know from your own experiences, this is a broad community with a lot of different activities. And so everyone has a role to play from collections to researchers, educators, aggregators. So where do collection-based societies fit into all this? And what I've done here on the left is shamelessly borrow a slide that Deb Paul uses with respect to introducing people to activities going on in our community at IDIG bio events. And what you can see here is it's an incredibly complex set of activities that are going on. And we don't even have the individual collections and institutions on this figure. These are all basically data aggregators and database efforts. And so trying to make sense of this, to me, is the job of the societies. So we have 3 societies listed down below: the Society for the Preservation of Natural History Collections, which for many years has been key in training and best practices associated with collection management and data management, as well as outreach. Then you have the Natural Science Collections Alliance, and we've long been involved in advocacy with the federal government. And that's one of the things that is absolutely required in order to move these initiatives forward. And then we're also involved with communicating what's going on to our members, and providing training for things like doing advocacy, such as how do you talk to your congressional offices, and about the needs for the broader connections. And we continue to develop ways to work on that. So coordinated and information associated with addressing these thematic areas going forward is one of the primary goals of the Natural Science Collections Alliance. And we do that through things such as organizing this particular meeting, which as Jyotsna Pandey, our new executive director, talked about at the beginning, is really important. And then finally, I wanted to mention the American Institute of Biological Sciences at the end, because I think they get overlooked far too much with respect to this. So we have all our collection-based work that is covered by the NSCA and SPNHC, but AIBS represents a large number of biological societies, and also a large number of institutions that do biology. And so they provide a place where our association allows us to talk about the things that we care about to a broader biological community. So for instance, associated with issues such as Nagoya, as Linda was just talking about, become really important. So that's the role of these organizations, in my opinion. Next slide, please. And of course, we have this challenge that we face with respect that, on the one hand, we want to represent large and small collections across the country, but we're also recognizing the critical importance of getting involved internationally. And so those are sort of 2 different ends of the spectrum. And we do that in various ways, but what I want to emphasize is that all of you are able to participate in these societies in one way or the other. And I want to really encourage you to think about ways to do that. Most of the people that have presented today from the natural science collection side have been participants or participated heavily in groups like SPNHC and NSCA over time. At the international level, we're working with groups like IDIG-Bio, GBIF, and DISCO. Joe Miller, the executive secretary for GBIF, is on the call today. And we've been discussing with them an upcoming series of consultations to discuss how we're organizing at the global level biodiversity data. And so we continue to look to ways to work more internationally, with the recognition that we need to do that not just with the developed countries, but with the entire world. Next slide, please. So one thing I wanted to emphasize, because it's come up over and over, is that digitization has tremendous benefits. And I wanted to provide one example of that from a project that I'm involved in, which is OVERT, which is an NSF-funded thematic collections connection grant to digitize vertebrate fluid specimens using CT scanning. And one of the things that I always like to emphasize to the public about this is, if we digitize our collections, they will be used. And this was provided to me by Doug Boyer and Julie Winchester at OVERT at the beginning of the pandemic, or as we were going through the pandemic. And what you can see here is that the downloads of these specimens is going up, but the 2 largest periods of download were periods after the pandemic started. So here we are at a time where we can't get into our collections, and yet if we digitize these specimens, these extended specimens, they'll be accessed by researchers in all kinds of times, and this is just an interesting representation of this. Next slide, please. Then the other thing I want to emphasize, which is, again, something that'll be apparent to everyone, but there are always more ways in which we can begin to bring this to the public and our colleagues in other biological sciences, is that the novel science possible is revolutionary. So on the left here is a study that just was recently published based on CT scanning of fluid specimens of kingfishers. And this is a group that has spectacular bill morphology related to plunge diving and insect feeding. And you can look at the variation in new ways using CT scanning. There's a paper that will be coming out soon related to this one, in which the bill morphology is compared to development of brain structure or to brain morphology and brain size. And where I'm going with this is we're beginning to reach out to fields such as engineering and neurobiology with respect to the capacity of work that we can do with these specimens through CT scanning. The example on the right is one from the Field Museum, where we've assembled a large collection of migrant birds, common migrants through the Chicago region that have been collected every spring and fall over the last 40 years. And this has resulted in this incredibly large data set where we can begin to look at broad trends in what's going on over a 40-year period in these vertebrates. And the pattern is that body size is going down, and that's consistent potentially with implications from climate change. And I just want to point out that, again, we always talk about what kind of collection possibilities there are across regions. And the truth of the matter is we need collections throughout North America to be regularly assembled in order to begin to look at trends more effectively. And because of the possibility of digitization, we can connect these specimens over time. Next slide, please. So again, I think that what you've seen over the course of these presentations is that it's an incredibly exciting time for our communities and where we're going. And these national reports provide blueprints for where we need to go. And it's not to say that there aren't challenges, and we're certainly concerned about what's going on with respect to COVID in our institutions right now. But there's a lot of positive upside with respect to this. And as things go on and we get a new administration in Washington, we need to be able to argue for the things that we need effectively. And that's where these societies like NSCA, SPNHC, and AIBS have key roles. And I'll turn it over to Barbara now.
So hopefully we've made the point. There are quite a few things to think about and to do in order to develop a coherent plan for an expanded specimen network, which is, in our view, one of the best ways that we can advance the use of biological collections for research and education. What to do next? Listed on the slide are some of the activities that the BCoN team is exploring. We're very much interested in enlisting a much larger group of biodiversity collections professionals, researchers, educators, and other data users to help in this effort. And one of the main goals of this program has been, we hope, to interest you all of you in the audience, in joining us on this journey. I'm very encouraged by all the thoughtful comments and questions that you've raised, and I'm hoping that means that you will be interested. So thank you very much for listening, and we look forward to hearing your thoughts.
All right. Thank you, Barbara, and all the other panelists for your remarks. And that concludes our presentations from panelists, and we will now attempt to address some questions from the audience. So we received several questions prior to today's program from registrants, so I will start with a question from there before moving on to questions posed today. So Mason Heberling asked, What should individual collections do now to foster the extended specimen movement? Barbara, would you like to take this one?
Yeah, it was a great question. And there are a number of things. I mean, first of all, continue to digitize specimens as you can, and not just specimens, but derivative data that you may hold as well. This could be in the form of images, notes, field books, tissues, microscopic preparations. It would be a great idea to imagine, considering the holdings that you have, to design a pilot project where you have holdings that could be linked together in a kind of a pilot ESN, if you will, just based on your own collections for starters. You could assess what parts of your collection you have, a range of documentation that could be shared, and you could begin to work on putting that together. Well, obviously, that's something to do as we gear up with all the proper identifiers and infrastructure in order to make this a broader effort. However, you could have a good project out of that, and you'd get a sense of the wide range of activities that are going to be involved. Another thing is to decide to take the lead in one of the areas discussed here today where we face implementation challenges. I use the example of Ramona Wall and her team, who, inspired by the Extended Specimen Network and other things, now has a major grant to look into identifiers. And I'm trying to remember the name of her project, it's escaping me. But anyway, it's a funded project which is biting off one of the tasks that need to be done here. So everybody's welcome to jump in and find something that they'd like to address and to do it.
Thanks, Barbara. There was another question which was sent earlier, and this was by Deborah Paul. What roles does standards play in moving forward the recommendations from the two reports, and what standards or protocols are missing? Andy, would you like to take this one?
Sure, I think standards standards are vitally important at various different levels. You know, the standards associated with the way that we catalog our specimens and the way that we portray them to the outside world is extremely important in terms of standardizing the way those, you know, the way that data is presented. But then there are also a bunch of standards associated with the way we integrate data together. You know, unique identifiers and those kinds of standards as well. So I think standards have a extremely important role to play here, but the number of standards is very diverse, and I think we need to drill down and look at exactly which of those standards are applicable and which ones we really need to implement in order to make this happen.
Thanks, Andy. There's a question regarding, you know, how far should the specimen be extended? And this is by Paul Kellerman. And he gives the example of genealogical and newspaper databases that can tell us where a person was, even if their specimens bear no collecting date. So I'm looking for thoughts from the panel on this.
So in other words, you would extrapolate where a person might have been based on other evidence, I'm guessing. Is that, you know, so a genealogical database might indicate where a person lived, and they lived always in the one spot, so assumingly the collections would go there. I mean, I don't see, I'm not sure I see how that could exactly be linked, although there's bound to be a record in a database, the genealogical database, that you might point to, at least in a note of some way. I don't know, maybe someone else has some different ideas about that.
Some of the stuff that David Shorthouse has been doing with Bionomia definitely falls into that kind of category, in terms of linking sort of collections people together, and some of the work that's being done on Wikidata to create pages for deceased people to be able to accumulate information about that person that can be then used to link them to records in databases. I also think that field notes have an important role to play there. There's a lot of information that is documented in these field notes that we all have associated with our collections. And some of that information doesn't necessarily make it into the database. So I think digitizing those field notes and making those field notes available may be able to give us an in into some of that information that can then be used to extrapolate some of this information that's necessary to link all this stuff together.
And the last thing I would like to add, so when somebody's doing a particular type of gathering of information for a particular sample, sometimes extra information is obtained along with the objective of that particular study, and that information will be useful to have it available too, because you never know what will be the unintended use of that information in the future. So one example is the early stages when people started sequencing and obtaining microbiome data from samples. Nobody knew that a few years later, it will be very easy to get all that data and find other organisms that were not originally looked for, that were present there, and that will give rise to a lot of more information. So there is really no limit. The more you can get, the better, because you don't know what can be the use for that in the future.
I can give one very simple example. If you know just literally the birth and death date of a collector, and you have them as a collector in your collection, but you don't have a data collection, you at least know when a start and end date was. So any kind of data can be used to infer and imply more exact information for your collection.
All right, great. Thank you all. All right, moving on to the next question, which was posed by Elizabeth Womack. For connecting living and specimen collections, that sounds like a great idea. How do you envision connecting all of the different parts that may develop to keep track of parts that are related to individuals? So if you sample blood from the same individual over multiple years, each blood sample may be different for the hormones and life stage, but same genetic sequence. And then trying it to offspring samples and then to the eventual specimen in the collections after it has died. Any thoughts from the panelists on this question?
I'll just say that it's a big, it's an example of one of those challenges that exists out there that we're gonna want to solve, and we're gonna need some thought put into it and what it means with respect to database management. But it's absolutely something that we should be doing, because the value of those kinds of that kind of sampling is really substantial given some of the things that we can do these days.
And to the other panelists, please feel free to jump in if there's anything you'd like to add. All right. So we also have two questions related to vouchers from David Shorthouse. Is there a recommendation for vouchering of heterogeneous samples collected for eDNA studies? Are collections willing and/or prepared to accept those? And the second question is, if depositing vouchers were to become a condition for a successfully funded research proposal, what must be done to ensure that high quality specimens using the best available preservation practices appear at the door?
The first one seems like it relates to living collections primarily, right? So maybe that's a Mansour or Curia question.
I was thinking about that. So many, for example, in the case of BI resources, sometimes we get NIAID to tell the research projects that they fund that once they finish their project, they should deposit the material in the collection. However, the collections do not have unlimited space or resources to accept everything. So then comes the quality or the usefulness of that sample. If you have 100 E. coli strains from very similar sources, they might have differences, no discussion about that, but there is a bandwidth that you have to be realistic. So you might not take all of those 100, and then you might have to narrow that to the most representative isolates of that particular set. And that also relates to the quality. When you get them into the collection, you always have to confirm everything. So you also check the quality of that material, and then you enter it into the collection. So that depends on the project and the situation where you have the samples coming in. So regarding to the way the sample is obtained and the quality and the standards to do that, that's something maybe that the funding and agencies have to keep an eye, because they must have processes for inclusion criteria into the study, and also the processes on how to obtain the samples. If they are not done properly, as a reviewer, when I get a grant for review and see, oh my God, these people have no idea how to collect, or these people are doing it very well, that already helps in the quality of the material, and that will also provide good quality on the science. So that.
I think biological collections are also starting to delve into eDNA. eDNA is becoming a huge part of biological collections. We haven't dealt with it yet, but there are all sorts of, strange things that go on with eDNA being incorporated into collections. Not only the fact that it is a heterogeneous sample that potentially has hundreds or thousands of species in it, but also the fact that there are all sorts of legal implications of, vouchering a water sample that you don't know what's in it, to then including blue whale DNA or panda DNA or whatever the case may be, and then having a sequence for that DNA, which then becomes part of the Nagoya Protocol and requires all sorts of permitting restrictions, et cetera, et cetera. It's a huge can of worms, and it's something that our collections are only just beginning to start to delve into, and I think it's going to become problematic in the future. In terms of David's second question about vouchering stuff, obviously that's why I think there is a huge importance to researchers and collectors interacting with the collection beforehand, before they go out into the field, in order to get that information from the collection as to how they should properly preserve the specimens, what kind of specimens the collection is looking for, what kind of data the collection is looking for, so that you can make sure that the specimens that are coming back are of the highest research quality and the data that's coming back is appropriate for those particular specimens.
Thanks, Andy. There's another great question from David. Are we envisioning a new digital infrastructure here, one that acts as a warehouse of links between entities elsewhere, or is it the responsibility of each individual collection to store and share links to the external entities?
I'll just say he might be better at answering that than a lot of us. But I guess what I mean, if you step back and think about where we were with respect to digitization 30 years ago and see where we've come today, I think partnering with all the other entities that are working on digitization and collection databases, which is something I think our community absolutely needs to do, where things are going to be in 25 years with respect to these databases is almost hard to predict, but a lot of these things ought to be possible.
And I think everybody has a role to play. Currently there's a lot of that work that's being done within the collection management systems of a collection. So I've spent hundreds of hours trolling the literature, trolling GenBank, trying to accumulate all of the references and all of the GenBank sequences that are associated with my collection. But there must be some technological way in which we can put that all together that obviates the social aspect of me having to spend hundreds of hours making those connections, and have some automated mechanism of being able to make those connections between publications, GenBank sequences, ISOBank sequences, whatever the case may be, and linking all of the stuff together. Whether that is a sort of combination of CMSs and aggregators and publishers and all of those kinds of things, or whether we come up with a freestanding solution that takes all of those different components and puts them into a sort of integration hub that then allows us to be able to integrate all this stuff together. So I'm thinking of things like Globi, where they're starting to integrate data together from various different disparate sources, host and parasite information and those kinds of things. If you think of sort of a Globi on steroids that would then be able to link all of this information together in a central hub and wouldn't rely on any one individual group or one individual collection making those connections themselves. That to me seems like the most sort of forward-thinking approach to this.
So just if I could jump in on that,
yeah, go ahead.
Follow on from Andy's point. This is just the kind of thinking that the NASEM committee is putting forward in this idea of what do you call it, an action hub, or any of the terms that Andy just used. It's this sort of resource and integration across a very complex community. I mean, John Bates's slide is just overwhelming in terms of the diversity of the different parts of this community, and I'm sure that's only a fraction of what's going on. So that's the sort of thinking that we had in mind in terms of a way to kind of get above the diversity, integrate it in a way that you can then go back out into the diversity and everyone can benefit from it. I have no idea what that would look like. There are lots of acronyms that Andy just gave us, that could be roots into that kind of thinking. But it's the kind of thinking that other communities, any of you have ever been to NCES, National Center for Ecological Analysis and Synthesis, they do this kind of work. Succinct does this kind of work. That's the Social and Natural Sciences and Environmental Sciences Integration Center. So there's kinds of thinking that's out there that's been funded by NSF. They fund these centers for about 7 years to get up and going, and then they're expected to develop their own kinds of funding. But there are models out there for that kind of thinking. It's also, if I could just put a fine point on it, it is the kind of thinking that also comes out of these decadal plans, which is the other phrase that's in the NASEM report. May not be familiar to everyone across the country or even the world, but they are these 10-year explorations by communities like the astrophysicists, like the folks who run the colliders, the big ocean vessels. They come out of these decadal plans which come up with recommendations from the community that goes to NSF and also goes to Congress and can get large-scale funding for big projects that can really benefit the community.
Go ahead, John.
I'll just point out that I was just trying to look for the program announcement, but the National Science Foundation has just put out a call for an open data environmental biology structure, I guess, where they're going to solicit proposals for $20 million for an institution to oversee some of these kinds of questions.
It's the kind of thing you might be able to write in on, John. Good thought.
There are two sort of connected questions. One by David Schindel. He asks, are there plans to develop an implementation plan that integrates the recommendations of the two reports? Yes, certainly. And a related question is, what is an example of a major milestone for these envisioned community efforts? In other words, when do the high fives happen?
When we have identifiers that are agreed upon across the community that we can put in our databases, that would be one. We've all agreed that there'll be identifiers not just for specimens, but for all the component parts of the specimen metadata.
And an implementation plan is definitely on the cards. You know, our thinking along these lines is we've put in a proposal to NASEM for a bunch of workshops to look at these 5 individual thematic areas to try and bring in as many community members as possible to discuss these and figure out, you know, what do we have? Where do we go from here? Where are the holes? What do we still need to develop in order to make this happen? There's also the GBIF discourse sessions that will hopefully start that discussion a little earlier and lead into these workshop proposals. And then all of this will hopefully lead into an implementation plan that we can then use to ship to NSF or any other funding agency to then look at putting the structures in place to actually start working on this.
Thank you. There's a question, anonymous question. Can anyone address possibility of automated or AI-assisted data processing technology already used or may be developed in the biocollections management, such as collection use data analysis using AI, smart contracting, and regulatory compliance using legal technology, etc.?
Most of the AI applications I know of are about matching, trying to identify unknowns, taking images of known organisms, specimens, and developing algorithms to match them to unknowns. However, when you combine data mining and vision, computer vision, the sky's the limit. So I think there are many, many more possibilities to be explored.
There's a question about, so the question is what this panel is talking about has neglected many of the systematic problems that collections have that were raised in the NAS study. Without solutions, many museums or collections will face limitations going forward. I include infrastructure, support staffing, etc. Any thoughts on that?
Well, I'll just jump in quickly and say that it's obviously we certainly aren't ignoring those. We all live those problems every day, but the bottom line is one has to address the value proposition for collections, and the best way to do that is to show how relevant they are to current research. And that may not solve all the problems, it might not make every collection solvent, but there's no question that demonstrating their value will be key to their success in the future. And we believe that extending the extended specimen network is one way that we can greatly increase the value and the utility of these data across a wide range of research and educational areas.
Mansoor, go ahead.
In the living collection area is even more important is not only how do you cope with that, we become the essence of that reclassification. So in the case of bacteria, all the species definition is relying on a single species strain that is called the type strain. Biological collections must have those strains, and they become the standard to define the bacteria related to that to classify. So collections are all the time keep obtaining those new species developed and described in the collections. And this is not going away. If you go to the historical collections, you go back to previously deposited material with one classification, you reanalyze the classification with current or novel technologies, you end up reclassifying materials that you have had for a long time. So they become an amazing source of information to keep the taxonomy current. And everybody is always complaining that the names keep on changing, but the taxonomy and the detail is even more and more specific. So the living collections are essential to do that. Viruses fall into this category, fungi, parasites. So it is not only an issue, it's essential for a biological collection to have those strains there and to keep on always reassessing how the material was classified in the first place. So it's never ending.
Right. Thanks, Mansoor. So there's a question by Hugo Gant. Is the future of small, sometimes historical collections scattered across departments or colleges with virtually no university commitment or support to becoming incorporated into larger collections of better supported institutions? And Anna,
I'll take that one. So I was actually typing an answer to it. That's why I would argue that that is actually not a good outcome. I mean, we don't want to lose the specimens because they have an inherent value, but they're often tied to local expertise as well. And when we run the risk of thinking of aggregating everything all together, we're also talking about influencing our workforce, decreasing exposure, and decreasing that very real interface between local communities and collections, both from a research perspective, so that has to do a lot with local knowledge, but also it provides a finer scale resolution. We have a couple of recent papers that came out that show the unique contributions of small collections. It's not just a duplicate of what you see in the large collections. Sustaining them also sustains the research and the ongoing collecting. So I'm not sure if that's the same answer someone was looking for, but I would say we need to do more to provide the basic support needed for those collections and find alternative mechanisms to support the curators and researchers working in them.
John, go ahead.
And I just completely agree with Anna, and of course, and I work at a major natural history museum. I mean, the value of these collections across regions is just essential, and they're used for teaching. They just have all kinds of roles to play. And so I think that what's incumbent upon us to do, and this comes back to societies, is continue to provide mechanisms and best practices with respect to helping people at smaller institutions that come into these situations be able to effectively articulate why they're there to their administrators. And it's something that it will continue to happen. It's never going to change, but I think that one of the things we can do is think creatively as a community about for instance, what's worked, and making those kinds of stories available to people so that people can begin to see what kind of things they can do to argue for their collections at smaller institutions.
I also think there's a lot of resources already out there to facilitate the digitization of the regional collections. And so there's scnet.ufl.org, I think. I might have that last part wrong, but if you look up SCNet, you'll see that there's a whole series of webinars that are talking about ways to advocate for and support regional collections, whether that aligns with your universities, and usually they're in universities, but not always, but what is the mission of your institution and how are you aligning with that? And I know in some places, the education part is what's gonna maintain the relatively limited budgets that they can operate on, which isn't optimal, but is doable. And I also know that there's a significant amount of literature on the educational value, so you can always like feed into that. But again, using those resources and working more collaboratively, like our community does really well, I think has made a big effort towards including those groups. I really advocate for SPNHC. They've had annual webinars. It's been a really supportive place regardless of the size of the collection.
And we're closely approaching the end of the program, so maybe we can address one last question from Subrata Gayan. Is there any training program available for collaborative taxonomic research?
In the living collection field, yes. You have the Global Catalog of Microorganisms. It's a huge project, and their aim is to sequence every single type strain of bacteria, and they're also starting with fungi, and they're doing microbiome analysis and samples. And the idea is to create a single database to unify all the taxonomy of bacteria. And this implies that the strains are available to researchers from the different collections. So all the information is gathered in a single place. So you can have all your IDs done using their material and their sequences. They're providing the sequencing for free to the researchers that do not have money, or the collections that cannot afford handling those analyses themselves. And it's an international effort. It's not just one country, it's all the world.
Thanks, Mansoor. There's another question, which if we can answer now, that would be great. Related to Hugo and Anna's comments, is there a centralized place to alert the community of small collections at risk or in need of rehoming?
I know that a lot of that is dealt with at the discipline level by individual societies and individual groups. SPNHC at one point was talking about a clearinghouse for orphan collections, looking at providing the structures necessary for people to advertise an orphan collection that potentially is at risk, and thereby allowing the community to come together and provide resources to potentially take over that collection or help out in any way that they can. I'm not sure where that ended up or whether it was actually put in place, but it's definitely something that we need to look at in terms of a sort of community-wide response to these orphan collections, and trying to ensure that they are maintained either in their current location or are taken on by another collection and looked after.
All right.
I'm seeing a couple of things that are coming up in the panel talking about something GBIF does with orphan data, as well as the Society of Herbarium Curators. I do think that your comment, Andrew, is really good about regionally people kind of take care of those, both by discipline and by within a region. And I've seen that be really effective in Michigan, as a watchdog that's sort of watching those collections, which we've been fortunate to have. And I think he's on here with Rich Rabler. And by keeping that eye on those collections at that level, it's helped kind of leverage things within the region. And it looks like Jennifer Zaspel is saying NSCA can also provide support.
Yeah, and I just come back to I guess there's some inevitability with respect to that, but I think the real goal needs to be trying to convince these institutions that if they're giving up those collections, they're actually losing something substantial with respect to what's disappearing. And if there's any way to get them to think about ways to maintain those collections and actually do some hiring that would actually use them, we need to continue to advocate for that and think of the best ways to get ahead of that, essentially, as those issues come up.
Thank you, John. Before we adjourn, I just want to mention that I've included a link to a supplemental Google Doc in the chat. This document includes some supplemental information about today's webinar, like references, links to websites, and so forth. And the document also has a space for each of the subtopics that we discussed today for comments and questions from the community. And this document has been made public, so please feel free to add any comments you have, or add any questions that we didn't get to today, and we will endeavor to address these on the Google Doc. And this document is sort of meant to serve as a mechanism to keep the conversation moving forward and to gauge interest and engagement from the community on the planned future efforts that we heard about today. So if you want, you can go ahead and add your name and email at the end of that document so we can keep you informed about upcoming events. And we will also share the link to that document and today's recording, as well as any resources that were shared over the chat during the course of the webinar today in a follow-up email. So keep an eye out for that, and please feel free to share that with those who are interested. And just to end, I want to thank all our panelists for a very productive and informative discussion, and a big thank you to all the participants for joining us and for asking such excellent questions. And a special shout out to all our international colleagues joining us from outside the US. Glad you all could join us today. It is very encouraging to see the robust attendance from the collections community today, and I certainly hope that you will all remain engaged in future discussions and collaborations on this topic. So thank you all, and have a great rest of the day.
A big thank you to you, Jyotsna, and AIBS.
And the
AIBS. Definitely look out for those GBIF discourse things that are going to be coming soon to continue this discussion. And hopefully some workshop proposals that are going to come out of BCoN that will help us continue this discussion too. Thanks, everyone.
Thank you.
Okay, thanks.